Welcome to Brain Ponderings, your window into the world of brain research with unfiltered conversations with experts in the field. Today it's my pleasure to welcome Professor Thomas Hartung. He's the Dorenkamp Zbinden Endowed Professor and Chair for Evidence-Based Toxicology in the Department of Environmental Health and Engineering at the Bloomberg School of Public Health, Johns Hopkins University. He's also director of the Center for Alternatives to Animal Testing. He hails from Germany originally, and he has a post kind of similar to what he has at Hopkins in many ways at the University of Constance in Germany. And he's doing some fascinating work in his own lab, but also in organizing around the world various laboratories and initiatives to to do multiple things. One is to advance technologies in what's called organoids. We've had one or two podcasts on organoids before, looking at brain development or other aspects, and then AI. And he's had a long interest in reducing the use of laboratory animals, mice, rats, non-human primates. And so we'll talk about these things. And he has a lot of really neat initiatives that we'll talk about. One is this Organoid Intelligence Initiative, which is trying to tap the value of these microbrain-like structures, organoids, and try to understand what's going on. We don't know what's going on in the human brain really at any refined level in terms of intelligence, for example. And then the other thing he's working on is merging the organoids with the AI with some neat new technology. So welcome, Thomas.
Thanks for having me, Mark.
Can you talk briefly about your background? You grew up in Germany. How did you get interested in initially science and then the field you ended up going into?
Yeah, I was infected by the science virus very early. Already at the age of 11, I had my own little laboratory in the basement of my parents' home. So I was the nerd kid, yeah, so, who was into any type of science all the time, yeah, and very quickly doing these national science competitions and things which were possible. So science was always my thing.
This was in the 1970s, late 60s, 70s.
Yeah, I was born in 1963.
Oh, 60.
So in the early 70s, I started to have my own lab and spent every pocket money I could get to buy to talk some of the pharmacists out of some chemicals, or
Ah,
did go
oh,
to.
interesting. And what were you applying the chemicals to?
I did simple experiments which were first quite impressive for kids to see, color changing effects,
Ah, I see.
smoke forming.
Yeah.
But I also always did chemistry and biology. So microscope. I
Ah,
used my pocket money also to buy a fancy microscope. So real one, not one of these kid ones. And
like,
I
like this one.
yeah. Mine was even more modern. But you get it. And I was so fascinated already at the time when I saw that there is a study which combines chemistry and biology, which is biochemistry. So I decided let me think, the age of 12, I decided I want to study biochemistry and identified very quickly there were only four universities at the time in Germany. Tübingen was the best one. That's why I want to go. And that's what I later did.
Yeah, that's a good place. I have a former postdoc of mine is a professor there now.
Yeah.
Works
No, I think
on
it's a wonderful
stroke and
historic village, I have to say. It is just 30,000 people and the university of 20,000. So
yeah, we went. went on a trip with my wife and kids to France one time, and then we rented a car. I think at Strasbourg, rented a car and drove through the mountains to Tübingen.
Yep.
Yeah, that was pretty interesting.
It's very
Okay,
nice.
so
Good.
then,
So then I also started very early on in 1976 to program. And the
uh,
parent association bought us a computer, a Wang computer, 4 kilobyte of RAM, and it stood there. It was $25,000, and no teacher dared to touch it. So I asked for the tutorial and read it at home. Then I started programming it, and so I had always also this idea of applying computers, using computers, and this was very helpful later now with the interest in AI. Yeah.
okay, but then there was kind of a gap, and so you had that in your background and your training and your interest in, and then you did a lot of work, toxicology research, right, for your,
Yeah, I did first study mathematics informatics while I was doing civil service. In Germany, we had mandatory military service in the early 80s. This was the only thing I could study at a remote university. So that's why I intensified. But then it was finally Tübingen in biochemistry. And after a year, I understood that I want to apply this biochemistry to medical problems. So I twisted arms with the university and got accepted in parallel for medicine when I had to drop mathematics and informatics. So I did not complete this. But I'm biochemist and physician, MD, PhD. And, and then within biochemistry, my later mentor created a new track beside animal and plant physiology. He started, Albrecht Wendel, um, uh, toxicology, pharmacology, and chemical analytics
I see,
as a third track.
I see.
And I was the first student of this track, and this was for me the ideal combination, doing in biochemistry something already which was more medically oriented. Um, and that's why I moved into the fields which are using most animals and I
Yeah.
was absolutely not into this. I was an animal lover, yeah, all my life. But pharmacology and toxicology, these are areas where we use a lot of animals and probably have also a good justification to do so. Um, but I was instantly spotting opportunities where I could work with cell culture or other tools.
Yeah, I started out with cell culture and then, we were doing a lot of work relevant to Alzheimer's, Parkinson's, stroke, and glutamate, neurotransmitter, right? You can kill neurons, process called excitotoxicity. But then, in case of Alzheimer's and Parkinson's, the field, the idea was you want to have an animal model that recapitulates the brain pathology and the clinical symptoms. So plaques and tangles and cognitive impairment in Alzheimer's or dopaminergic neuron degeneration and so on. But I was always I didn't do a lot of hands-on work with the animals. I same as you, I'm an animal lover, and I don't like the idea of animals being put through these things if they absolutely don't have to. So I think the work you're doing is really important in that area. Um, maybe at the end, we can come back to maybe you're going to talk about what can be accomplished without using laboratory animals. At the end, maybe we'll come back and talk back and forth about are there areas that we really still need the animals for? I guess the idea would be to minimize the number of animals that are needed to accomplish.
But
Okay.
that's the essence, yeah.
Yeah,
Uh, I'm happy to endorse animal tests which are done as humane as possible if there is no alternative.
yeah,
Yeah. When
yeah,
I'm a physician in the leading school of public health, I really want that we can treat people, that we can prevent disease.
yeah,
But I have to say, over the decades, I learned there's very, very little defendable animal research. There's a lot of animal research which is not worth the paper it is printed on.
Yeah. Yeah. I tend to agree with that. And that continues. So okay, so, I used to work in two-dimensional cultures, essentially, take embryonic neurons from embryonic rat brains, put them in culture, they grow axons and dendrites, they do form synapses, and they do become electrically excitable, and we did calcium imaging, and you can see spontaneous activity, but um, we didn't have many glia in the cells. There's no myelination. We actually tried to reduce the number of glia, so because it can complicate some of the interpretation if you're doing biochemistry, right? Uh, and then also, though, when I was a postdoc, I'd shown in these 2D cultures that glutamate can promote the formation of synapses between neurons. And I wanted to know if that occurs in vivo. But weren't doing any in vivo work. So I did what's called explants, brain explants. So I took chunks of embryonic or early postnatal, actually it was hippocampus, and then another chunk of entorhinal cortex, which is a brain region that innervates the hippocampus. And this explant, and put it in the culture, the explant of the entorhinal cortex would grow axons across the bottom and then actually form synapses with the hippocampus. So it's kind of getting more towards an organoid-like thing, but with a more crude approach, I guess, at the time. Can you talk about, well, let's talk about organoids first. What are organoids? They're three-dimensional. What's the technology for preparing them, maintaining them, and then the various technologies that are being developed for interrogating them, their structure, their function, their, yeah.
Yep. Okay, it's a bucket full of questions. Yeah. The you know, most cell culture has been done in type of petri dishes, yeah, flat, 2D, but our organs are not flat. The cell density of a cell culture in a petri dish is about 1/1000th of a tissue, um, which means for nerve cells, for example, it's very difficult to form the same amount of connections of synapses. Um, it is also different in many other aspects. The one of the aspects which is very different in cell culture is that we don't have homeostatic conditions. The conditions are permanently changing.
Yeah.
We are adding fresh media at one time point, and then there's abundance of nutrients, of oxygen, and everything is consumed very quickly. Waste is accumulating. Then in a second, we are changing this again. This is terrible for cells. And there's a third component which is really terrible for cells, which is especially for neurons, we are boring them to death. They have nothing to do. They have nothing to think about. And these cells are longing to be active. They're looking for, isn't there something where I can get some input? They're growing out, trying to establish connections, and they start spontaneously communicating with each other. So all of this is necessary to really come to a complex in vitro system which reflects some functionality and architecture of an organ.
Yeah.
And that's what we call them now microphysiological systems. Because they're micro, they're small, just a functional unit, but they're physiological, they're expressing organ functions. And organoids is one type of these. Typically, organoids are started from stem cells. So we allow essentially the embryonic development to take place. And they form spheres. They form some type of three-dimensional structures and self-organize. That's what organoids in particular are about. Um, in case of the brain, that's quite easy because human stem cells long to be neurons, and you don't need to do a lot. You add a few growth factors, and they start forming brain tissue. Yeah, that's a it's really fascinating.
And then, so in the organoids, they're spheres. They have a limited diameter because there's an issue of diffusion of oxygen and then maybe even nutrients can only diffuse so far. That's why our blood vessels are and capillaries are, going all over real close. But and so then and then you mentioned about in the static 2D cultures, you change the culture medium every few days. Every time you change it, you're adding medium that has uh, way too high level of glucose. It's like diabetes like levels of glucose, high levels of amino acids. So you're getting kind of a constant activation of the growth state. The mTOR pathway, for example, is probably going up and then back down and then up and down. The pH is changing. We have uh, a pH indicator in the medium, so it's usually kind of orangish, maybe a little pinkish, and then over time it gets yellow, it gets acidification, right? So that's very abnormal. So the organoids, somehow there's a perfusion system to get kind of a constant physiological levels of nutrients, oxygen, and so on. Is that how that works?
I mean, I would say the most physiological cell culture systems have some type of perfusion to allow
Yeah,
the constant exchange of media, so that the cells can arrange and ex know what they have to expect.
yeah,
As you rightly said, the phenol red, which we add to cell culture media, is turning yellow, which actually means that these cells are not really having enough oxygen or not using the oxygen.
yeah.
They're using anaerobic metabolism, which leads to lactic acid, the stuff which gives us muscles after exercise, and the and they don't because they either don't get the oxygen. Um, a cell culture is consuming very quickly the dissolved oxygen, and then we have no red blood cells to facilitate the transport.
Yeah.
From a cell perspective, the cell culture media are of extremely high levels of fluid separating them from the oxygen. So they really very quickly move into anaerobic metabolism. And we expect them to perform normally under these conditions. So there's a lot of aspects where our primitive cell culture, which was the dominating or is still the dominating cell culture, um, is at the moment being refined. Um, it also has to do, for example, with cell sourcing. We used for a long time predominantly tumor cell lines when we are working with
Yeah.
humans. Because the only thing we can get. I'm working on the brain as you have been. If I want to pick somebody's brain, and I don't mean it literally, because I say, "Can I have a bit?" I will not get a lot of friends among the people I ask. Yeah. Um, so it is really sourcing these materials, and all of these problems started to find solutions over the last 20 years. Um, you know,
When I was at NIH, one of the senior investigators in my group was Mahendra Rao. I don't know if you came across him, but this was back early 2000s, and he'd been doing a lot of basic research on human embryonic stem cells.
Yeah.
And then I recruited him. He got the lab going, and then Bush came in as president and put a moratorium. But then after he went out, Mahendra got recruited to NIH to start but he had developed some methods for kind of amplifying the human embryonic stem cells with the idea of, using those. Uh, well, I think there still are labs doing it, but now with the induced pluripotent stem cells, I think most people are going to that.
Yeah, these were really enormously important breakthroughs. And they are just dating back some 25 years. Jamie Thompson developed the human embryonic stem cells,
Yeah,
which really became available in 1999.
yeah,
And this was, in principle, the breakthrough. The problem was that they used surplus embryos from in vitro fertilization to produce
yeah,
them.
yeah,
So to be clear, if a couple um, goes through in vitro fertilization, a lot of these embryos are being produced, and typically three of them are implanted. The
yeah,
rest is frozen away, so that next time, if there is a second shot or third shot necessary, you just thaw them and use them. After the desire to have children is satisfied or the people give up, not another shot for 20,000 plus dollar. Um, it is it is very often that they decide, yeah, instead of throwing these away, we they allow research to be performed on this. And
yeah,
this was the material on which, with the consent of the donors, stem cells have been produced.
yeah.
But for religious reasons and others, some saw this as ethically problematic. And we had discussions about this. Can we do? I succeeded to get the first European project to fund it, which was using these type of cells. Um, but very difficult. A lot of um, loops to jump through. Yeah.
Yeah.
And this all was resolved when Yamanaka in 2006 described how to produce a cell like an embryonic stem cell, which was generated from a piece of skin. He got the Nobel Prize in 2012, one of the fastest ever, just in
Yeah.
6 years, because it was such a fundamental thing suddenly to have ethically, reasonably clean cells available with the same potential to produce any type of tissue.
And then many other labs have since developed methods to direct those pluripotent stem cells into whatever you want, pretty much neurons, cardiac cells, glial cells, and so on. So it should have got a Nobel Prize quickly, right? Because it's
I'm for it. I'm rooting. No, I did. I mean, this was really a technology which obviously overcame a big hurdle for the broad use of these cells. And
my most recent podcast was with Magdalena Gutz. You
Mhm.
must have come across her at Munich.
Yeah.
And she's developed methods to directly convert astrocytes into neurons with the idea that maybe after a brain injury, there may be a way. Typically, the astrocytes don't die, even though neurons around them died. So the idea is, can you introduce a few genes or whatever into these astrocytes and convert them to neurons to build new circuits?
I mean, they have a common precursor, the neuronal precursor cell.
Yeah,
And this is, let's say, opening up for conversion as the reprogramming of the skin cells uh, has shown.
yeah,
Yeah.
yeah.
And they're the only cell which are proliferating in the brain. So for this reason, it's also for, let's say, um, a repair idea, certainly a good starting point. Yeah.
Okay, what's the getting quickly back to the trying to reduce the animal use. So I mentioned in introducing you that you're have an endowed chair from this Dorncap Zbinden Foundation. Is that right? What is that foundation, and what are its goals?
Dernkamp-Sabin Foundation was created by the late toxicologist Gerhard Sabin in Switzerland, a very, very renowned toxicologist who early on questioned some of the approaches we are taking. Particularly, he was critical about dog use in toxicology. So
Yeah.
there's nothing gained. And Hildegard Dörnkamp, a layperson with no scientific background, but who was affluent enough to put in her lifetime about 30 million into alternatives to animal testing. And I'm very closely associated now with with the association. I'm actually the president of the foundation now. But the foundation took at some point before I was associated the choice to create in total 5 chairs worldwide to promote into education and into curricula the idea of new technologies to replace animal testing. And I was when Hopkins offered me a professorship and to direct the Center for Alternatives to Animal Testing, one of the premier institutions in the world in this field, created in 1981 by Alan Goldberg, my predecessor. When he retired, they were looking for somebody and came to me. And then Hildegard Dörnkamp said to me, Thomas, what do you think if I endow this as a chair? And because we were already having 2 others and we were later creating 2 more, and I said, sure. So this was a very easy negotiation here in Hopkins because I came and said, if you convince me to come, I bring the money for the endowment of a chair. So it was a
And
very
when did you move to Hopkins?
in 2009.
Okay, and then how did you, was it at Hopkins that you got exposed kind of directly to the organoid technology? Or was it before that?
I mean, I was interested in vitro systems, cell culture
Yeah.
systems throughout my academic life. I did my share of animal experiments in parallel, but I already started in my PhD to work with cultures, co-cultures of different cell types, in this case of the liver. I shortly after started to get interested in perfusion culture. And in 2002, I did change. I took the lead of an institute in Italy for the European Commission
Hmm.
for the validation of these new methods. And I had a postdoc who started working with brain organoids, at the time, rat brain organoids.
Ah,
And 2 of her PhD students joined me then in 2009 to build up a laboratory here when I started in Hopkins. And we brought in these rat organoid
I see.
brain cultures. And the first thing we said, let's humanize them. And we took advantage of the rich ecosystem here in Hopkins of Kennedy Krieger School of Medicine
Hmm.
and brought these induced pluripotent stem cells in and very quickly developed a human model. And we were beaten really just by about 4 months the by the publication of the first brain organoids in 2013. But we were then in 2016 the first to mass produce brain organoids in a standardized way, which had a lot of impact for pharmacology, toxicology.
Okay, so for pharmacology and toxicology, when you think of that, or I do, I think of kind of two things. One, environmental toxins, but two, testing the toxicity of potential drugs, right? Because you don't want something that's going to cause damage. So how can organoids, I guess, refine and accelerate toxicity testing?
I mean, you have to see an organoid a little bit like a living biopsy. Um, so you get a piece of tissue off from a person as if he had taken it out of the brain. And this is a fantastic opportunity. You cannot probe the brain, but you can produce thousands of brain organoids for a given individual. This has a lot of opportunities also for personalized medicine, for diagnostic
Mhm.
measures. You can in principle try therapies on this individual and personalize the approach. Um, for us, the opportunity is also to identify is there chemicals which are perturbing these developments. Um, so one of the driving forces for our research, why we wanted these brain organoids, was the enormous increase in autism.
Hmm.
Um, in the US, we have the best statistics thanks to the Center for Disease Control. Um, in the 70s, 1 in 10,000 children was diagnosed with autism. The last data from 2023 say 1 in 36 is now being diagnosed. And while in the beginning this was a lot of awareness and change in diagnostics, it is now over the last 10, 20 years, it is clearly an increase in the number of children affected.
Yeah.
And genetics does not change that fast. So there must be lifestyle factors, exposures which are playing a role, but nobody is able to really identify the culprits.
There was some, before we went live, I was talking to you about intermittent fasting, and so I've had a lot of interest in, as many people have, the obesity epidemic, and there's been an epidemic of maternal obesity and diabetes as well. And there's some evidence that a child born to a woman with insulin resistance, obesity during pregnancy, which is kind of an inflammatory environment for the developing fetus, those children have increased uh, risk for autism. So there may be a role for that, but we don't know for sure, I guess.
No, inflammation is certainly a risk factor. You can show that um, if the mother during pregnancy is having infectious episodes, that this is a risk factor. There's others. Age plays a role.
Hmm.
Couples are getting older and older nowadays, and especially interestingly, the age of the father seems to be some risk factor. But each and every component is contributing something. But we don't really have. This is the big thing we need to address.
Yeah.
But we also because we don't have an experimental system to do so.
But there
Uh,
does seem to be like an accelerated growth of the brain, if, if I'm understanding right. During brain development, seems to be more rapid proliferation of stem cells and kind of a, you know, accelerated growth. I think overall brain size is actually on average bigger at birth than someone who develops autism. So
there's a lot of different suggestions, but the causal part, yeah,
yeah,
is the problem.
yeah,
And we know that some chemicals produce defects in neurodevelopment,
yeah.
um, but we would love to have a
Uh,
system of testing it. And here
yeah,
the problem of the animal experiment becomes really evident. There is one protocol developed by the EPA and then later accepted by the OECD, how to test whether a chemical is producing effects on neurodevelopment. This guideline um, does consume more than 1,000 animals
mm,
to test this chemical. You pay about $1.4 million, and it takes you about 2 years to get the results. And it does not find stuff. Yeah. The APA reported once that they have looked into the last 200 studies which had been submitted to them. So it's about $300 million spent, yeah, and nothing was identified which was not allowed to go to the market.
mm,
So it is. This was the situation in around 2005. And that is when my predecessor here in Hopkins, Alan Goldberg, and me at the time working for the European Commission Italy teamed up and we said, we need to find a strategy how to test for this without animals. And we have done 9 international workshops and 5 international conferences and really formed a community to do this, um, which and the OECD has published last year, actually in 2023, yeah, not just 2025, the first guidance how to use cell culture systems to test for these things. So this is really a tremendous success, a 18-year story to develop from zero, a blank sheet of paper, um, a consensus that a set of cell culture experiments can help you to identify these substances.
yeah.
And for
Yeah.
us, it drove us into using the brain organoids as the most promising test system which we could identify in this process. Yeah.
Well, now I don't want to get into any, kind of cherry picking, but what about microplastics? Obviously, that's in the news a lot
Yeah.
lately. And is there evidence that the developing human brain has microplastics?
You can find some, and there is, I would say that there's a concern, but there's clearly no evidence.
Okay.
And you can identify in almost any cell type of the body microplastics, but many of them seem to be very inert, and it is single cells showing these plastics. It is not that every cell of the brain is
Oh,
impacted, but they could have inflammatory components. It is a new concern. It is addressed in more and more studies. But it also does not seem to be really something new which would explain this dramatic increase, for example, of autism over the last 20 years.
yeah.
Plastics are around
That's
much longer, though increasingly used. Yeah.
okay.
So it's
Then,
sorry, it's really about being able to follow such an idea, such a hypothesis. It's microplastic, it's a pesticide or whatever. You need a test system.
yeah,
And after the animal test disqualified, not only for ethical reasons. I mean, some people would not bother to use 1,000 rats or more, but it disqualifies for economic reasons. It's too expensive. You cannot use it for screening.
yeah,
2 years of delay getting to a decision, you cannot delay a drug development by 2 years.
yeah.
One day earlier to the clinic means about $1 million for the pharma company. So it's not possible
That
to.
makes a lot of sense. I mean, it hasn't worked anyway. That preclinical models of disease, it's easy, papers showing that, drug X is beneficial in animal model Y of Alzheimer's disease or a dime a dozen. But, not, it's in the case of neurodegenerative disorder, it's pretty much nothing that has been published. It's published in animal model, it has beneficial effects. Then the clinical trials uniformly fail. So that's clearly not working.
The failure rate in human clinical trials of things which were wonderful, all of them in these animal models, is in neurosciences, neurology, 97% roughly. Yeah. So they're not good predictors. Yeah.
Yeah.
And this is why there is a tremendous need for human models, and this is what these human brain organoids and brain on chips type of systems are promising, that you get a reliable tool into your hands. And what we then now put on top of this is that we said, wouldn't it even be better if we have a functional endpoint, which means if we can test the brain function, not just the structure, the forming of the brain. And this is what organoid intelligence is about. So you have to think, what is the cognition is the function of the brain. But it sounded thinking brain cells, yeah, that's impossible. But when we started to present on our brain organoids mass produced, some people said, "My God, are they conscious?" Because I showed they are spontaneously exchanging electrical signals, electrophysiologically active. Yeah, and then I easily said, they are thinking, but they have nothing to think about. They don't have input and output. And during the pandemic, when we were in lockdown, my wife and me, we were discussing. She had taken over by then. She's professor too in Hopkins here, Lena Smanova, and she has taken over our brain organoid research by then. And I had over the last 10 years specialized more and more on AI, linking to my early work in programming computer science. And we said, can't we bring them together? Can we have AI talk to brain organoid, like AI is now talking to whales or other animals, and see whether we can establish a communication. And this is what we called organ intelligence, OI. Um, and this idea has really sparked a lot of activities since then.
Okay, so then to do that, you have to have you have to be able to record electrical activity from the neural networks in the organoid. And then you have to, you have that output then that's coming out. And then somehow you have to merge that with a computer. And you can go the other direction, I guess. You could have the computer uh, put electrical input with different patterns, frequencies, whatever, into the organoid. And then, um, yeah, and then maybe the circuits in the organoid would learn from the input, and then the AI would learn from the output. Is that the idea?
Yeah, exactly this.
Yeah.
I mean, you describe two different experiments. One is what we call an open loop experiment, um, where we just feed signals into the brain organoid, and we observe whether it is reorganizing in response to this
I
and
see.
changing its electrophysiological behavior. You have to imagine that a brain organoid starts with random connections, and by feeding it information, we accelerate that it is moving from random to ordered system. We call this criticality. But this is a very limited set of experiments. Really fun it becomes as soon as you do this in real time, and you take the response of the brain organoid and translate this into an action. And then you give the brain feedback on the action. You achieved something. And here we really borrowed from our collaborators and partners of Cortical Labs in Australia. They were the first to suggest simple computer games as the representation of the brain's activity. They did choose the computer game Pong, where you have a paddle and need to keep a ball in like in squash within the field. Um, and this was a big show. When they published this, a lot of people got inspired by this work. And we have been working, and only 2 months later, we published then also including them as the concept of organoid intelligence. And Hopkins has been extremely generous in financing us with a lot of internal funding in the Surpass program and in the Discovery Grant program, so that we were able to develop our own, we call it Mind Gym. So different types of computer games our brain organoids are being trained on in order to show whether they improve the performance of these little tasks.
And then do you have people then taking the organoids trying to just is there some structural manifestation of them learning, or there's LTP, right, long-term potentiation.
Yeah.
That probably has to be occurring somewhere in the synapses.
Yes, it does. And but the I think was we also see the induction of the early genes of learning.
Ah,
So you can first see that there is a cellular mechanism which is started. Um, but we first have to overcome also some technical problems. You need an EEG for the brain organoids. Not only an EEG to read, but also to communicate. So we are following two different pathways here. We use commercial two-dimensional microelectrode arrays where we plant our brain organoids on. They have in the meantime 26,000 electrodes, 1 micrometer apart. So they're really good, yeah. But we also created with David Gracia's group here in biomedical engineering shell electrodes, which are really folding themselves around the brain organoid. And at the moment, we have at least 16 electrodes, which we can use for reading and stimulating.
So that's some flexible plastic type.
Yeah, these are materials, gels, which are changing their tension, and for this reason, closing and embracing when you add cell culture media. Um, well, beautiful bioengineering work.
Do these do these organoids uh, maintain circadian rhythms?
Oh, we don't know,
You don't know.
but this will be an interesting one. We are really um, I'm we are really doing these experiments at this very moment. Yeah.
Okay.
We have a we have a paper under review at the moment where we show that we have the machinery for long-term learning, that this is all working, and we are trying at the moment to identify um, ways of benchmarking this learning in a reproducible way. Because then we can start with patients type of things or adding chemicals to it and characterizing. But we also, our brain organoids can only be half a millimeter big.
Yeah.
We discussed earlier this problem of oxygen and nutrients coming to the core of these organoids, because they're rotting otherwise. Yeah,
Yeah. Yeah.
a rotting brain you don't want to train. We cannot expect it to be a good player of a video game. Um, so we are also building larger systems which have this perfusion, so the equivalent of vasculature of the brain. And our current development is towards a model of almost a centimeter, which would be already twice a mouse brain in size. Yeah.
Yeah. Wow. Yeah. That's
Then
great.
it's worth training, but there's a lot of challenges in this. Um, the same challenges as Elon Musk's company Neuralink has to get a permanent connection of an electrode which is implanted in a patient, which is not moving when there's vibration, which is not because if you lose the contacts, everything is lost. Yeah.
Yeah.
Or if there's a reaction of the brain tissue to this material, everything is lost. Yeah. And we are solving exactly the same problem but in tiny, tiny, tiny mini brains. And but at the same time, adding perfusion to it, it is fascinating engineering work.
Yeah. This is really fascinating, the organoid intelligence. And you had uh, and Frontiers in Science had a big, well, coverage of this, I guess you'd say, that this Baltimore Declaration. You had a meeting in Baltimore
Yeah.
uh, where you kind of came up with a synopsis of everybody's input.
Yeah. I've been working a lot with Frontiers, um, because it was one of the first open access journals publishers. So they have every article published is available in full to everyone for
Yeah,
free.
yeah, yeah,
That's really important. I was the founding editor of Frontiers in Artificial Intelligence, and over the last 6 years, we have published 1,200 articles in this journal, um, and so I did know them well. And when we developed, and I was part of this, the idea of a flagship journal for this publisher, Frontiers in Science, um, this became the very first article for this
oh,
new journal. And we have delivered. And in the 2 years since it was published, exactly, the article has already been cited 178 times, which is really
yeah,
very, very good
in
for.
2 years. Yeah.
And this journal is publishing very select articles, and is accompanying them with a handful of editorials, a kit version, a laypeople version, a graphical material.
I just wrote in an editorial for one of the articles that
Good.
they're going to have come out pretty soon.
Yeah. No, I'm very happy with this way of publishing, I have to say. And it is really making this a very comprehensive thing. If you show also different views, comments on
Yeah,
these, yeah,
yeah,
give access to materials. And in our case, we already did carry out the first workshop you mentioned on how to form now a community for OI. and also developed a declaration towards the exploitation of organoid intelligence. So this was a very, very comprehensive package which was formed here. Yeah.
All right, so let's go back to so what can't be done with the organoids? So I've been thinking about this a little. So you can artificially have sensory inputs, I guess, but I guess in theory, maybe it's possible to have the inputs very closely mimic the inputs from the eye, the ears, the olfactory system, taste, whatever. Then behavior. We did a lot of behavior in the lab, learning and memory, testing for anxiety, depression-like behaviors, social interactions, and so on. I was at the National Institute on Aging, so lifespan studies, which is uh, probably not so easy. I could determine an organoid lifespan, but I don't know how that relates to an actual animal. And there may be, well, the other thing is, I think it's really important to get, multi-organ systems. So, you have because you don't have a circulatory system, which means you don't have hormones. There's a lot of circulating factors that are very important, neuroendocrine systems and so on. So what's going on in terms of like linking brain organoids with heart organoid or I don't know, an endocrine organoid, something like that.
Yeah. I mean, all of this, what you described, is somehow in its infancy starting. Yeah, because it really has sparked a lot of interest. Not only more than 600 press hits from Financial Times to Wall Street Journal, Forbes, CNN, and BBC, everybody. Yeah? And we regularly also have television, radio, and these type of things happening. And this created also a community of, at the moment, about 500 people who are receiving our newsletter, who are contributing to to these endeavors. The users are at very different levels. The first ones are really about trying to understand the cellular and molecular mechanism of learning.
Yeah.
So things where we at the moment poke electrodes into macaques' brains and have them carry out simple experiments, you can do in principle with these type of setups. There's then a lot of research towards brain-machine interfaces. So can we really evaluate electrode materials in these systems? And I'm, as a pharmacologist, toxicologist, I'm very interested. Can we start developing disease models so that we can possibly identify drugs or can we identify toxicants which are impairing the function of the brain? And we have really some first steps. We did choose, for example, domoic acid, which is a shellfish poison
Yeah.
from algae blooming, because it is a very interesting substance which is directly acting on the brain and producing dizziness and even fatalities.
Yeah.
So we study the effects of domoic acid on the brain as a disease model. Yeah.
I have a section in my book on glutamate on that I talk about that incident up in Canada where they ate the shellfish that was yeah. So
And perhaps if I just can complete, sorry,
yeah,
the fourth level is then, and this is what the bioengineers are dreaming of. The brain is a fantastic computer. Um,
yeah.
it only in June of 2022, the first supercomputer exceeded the human brain's computational capacity. But this was a $600 million computer on 6,800 square feet. You can imagine the enormous installation which took place here. And that's 1.4 kilogram we are carrying on our shoulders. Yeah. So they want to learn how to build better computers. Whether this involves brain cells or just uses the principles, neuromorphic computing, that's a different story. And what you're describing of combining this, uh, we have active work or at the moment towards combining brain organoids with retina organoids so that we can
Yeah.
have an input organ, for
Yeah.
example. So we are also very much looking at the moment, can we give this brain a reward center? Because for learning, the most important thing for us is to tell it you did well. At the moment, we can only penalize. Uh, but we are looking for options in really making different active zones of the brain that we can replicate what is happening in the brain, that we tell the brain cells who did the work, good job. We get a little bit of dopamine now, and you're happy. Yeah.
They can, yeah, and then you can make them addicted. You could have organoids that are addicts,
Yeah.
right?
Uh, all of this is the limit is the sky. Yeah.
Yeah.
There's a lot of things, screening chemicals, de-risking chemicals which go, let's say, in pediatric clinical trials. Yeah, you don't want to impact on brain development, so I would recommend to test on this. Yeah,
Yeah.
Um, we know that also. I mean, these are very embryonic structures, these brain organoids. But astonishingly, if you take cells which come from an Alzheimer patient, they show some of the hallmarks of the Alzheimer brain.
Yeah.
So we already work here with our colleagues in the Alzheimer Center in Hopkins to and build at the moment brain organoids from Alzheimer patients in order to compare. Are they learning worse? Are they less plastic in rearranging functionality to respond to input signals?
Is that from late onset Alzheimer's, sporadic, or from early onset inherited, or
Um,
both?
we are doing both
Yeah.
because we really try to have a battery of these available.
Yeah.
It does take us typically between 6 and 12 months to get a cell line to produce brain organoids. So we
Yeah.
have initiated this with
A lot of work.
I think at the moment some 8 Alzheimer lines. We also produced cells from amyotrophic lateral sclerosis, another project, and in some rare diseases which affect cognitive functions in children, um, SYNGAP and leukodystrophies. So we have a broad variety of disease models. With our autism center here, we also follow up work with cells from autistic children. Um, we even introduce risk genes from autism into healthy genomes so that we can study individual genes. Um, and we
Yeah.
hope that we can enhance this research soon also by making organoid intelligence one of the endpoints to show then what the impact is. Yeah.
And what are your readouts in those? Depends on the model in terms of what are you actually quantifying?
It it really depends. The in in now at the moment where organ intelligence is not yet a standard parameter which we can measure on
Right.
scale,
Yeah.
we very often use metabolomics, for example.
Hmm.
So we're interested in the balance between GABA and glutamate,
Ah,
which is typically disrupted in these children,
yeah.
and some other parameters. And we have used this to show that what is happening in the brain organoids, if you have a toxic substance, which is a risk factor for autism, or if you introduce a gene, which is a risk factor for autism, you see the same derangements and perturbations which have been described for autistic children.
So yeah, it seems like so the GABAergic neurons, the inhibitory neurons, have a very high metabolic rate because they fire a lot, right? They're very active compared to the glutamatergic neurons.
Yeah.
And there's a lot of evidence that problems occur in those GABAergic neurons before glutamatergic neurons, even in Alzheimer's disease. And so you get this excitatory imbalance. And I think aren't kids with autism, they have increased chance of having seizures, I think. And
There's some, we are looking, for example, into SYNGAP, which is one of these
Yeah.
rare diseases. But we have the specialists here, and with some 40 cases. And interestingly, there's some children which have nothing despite the genes.
I see.
Others have autism, others have seizures, and some have autism and seizures.
Hmm.
So we're trying to use our systems to get these things apart.
Yeah, that's really good, really important work. And then lastly, uh, it seems to me that the ethics of using organoids is it's much more ethical than using non-human primates or even rats and mice. Although you mentioned that maybe these organoids have intelligence, so then you get kind of like almost philosophical question of can an organoid experience suffering or something like that? And I don't know. What are your thoughts on that?
I mean, we have instantly identified this as a critical problem, and we teamed up here with the Berman Institute for Bioethics. um, Jeff Kahn leading it, but also his associate Lomax Boyd, who is working very closely with us. And we said these type of experiments have bring in a new ethical component. At least theoretically, there could be at some point caution. Caution. There could be at some point self-awareness, consciousness, yeah, and suffering, as you rightly said. So we agreed on an approach which we called embedded ethics, where they're sitting with us and every week when we're discussing our experiments and listen in, they come to the lab
Hmm.
to see what we're doing. We're even discussing whether we could create a type of experimental ethics, where you ask what is needed to come to what complexity is needed to assign these properties. Um, we have just published a call with our Australian colleagues on the need for clarifying the terms we are using. What is intelligence in the context of an organoid? What would be suffering? What would be self-awareness?
Yeah.
And we are hoping that we can move the field also in this direction. And these ethical discussions are critically important. For me, when I'm trying to understand something, I always write an article about it. So one of my articles last year was really about the ethical, legal aspects around this, um, because I want to make myself aware of what the consequences
Yeah, I
are.
guess you could make the case. So you're taking a person, say, skin fibroblast, you convert it into a pluripotent stem cell essentially, and then you're growing an embryo in the form of a neurosphere or you're directing it in certain ways maybe to become uh, brain-like,
Yeah.
right? Um, and then, but it doesn't have the input from the environment necessarily, or at least not major input. So it's kind of like a developing embryo in the womb to a
Yeah.
certain stage. And then you get the question, well, abortion's legal and well, it depends where you are in the United States now, I guess, but to a certain age of gestation. So it's kind of the same ethical, in a way, consideration there, I think.
Yeah, I mean the with our tiny little mini brains of half a millimeter, I'm not really concerned. Yeah.
Okay.
Um, these are about 50,000 to 100,000 neurons,
Yeah.
which is, I would say that the housefly probably has that many. Yeah. And, but I still think we need fundamentally to clarify, is there a limit? And should there be a limit set? For
Yeah.
example,
Okay.
as you said, we could say um, 4 months is the maximum of maintaining these systems, or but because otherwise, at some point, you might come to the conclusion it is unethical to stop feeding them, yeah, because they have learned something. But also, what is the relationship of the donor to the system? Is the informed consent, on which we are working, obviously, we got these cells to do our work, but have we informed them that we are developing systems which are sensing, systems which are digesting information? So we need to understand what is necessary legally also to handle this properly
Yeah.
before we have systems which are capable of doing it.
Yeah. Yeah.
And the one of the things the ethicists at the moment are doing, they are talking to a general public, describing the work and asking more or less, when do people say, "Oof, perhaps this is too much. Yeah, you should not go there." And does this change when you say, "Yeah, but we're doing this to find an Alzheimer treatment, or
Yeah.
we try to prevent autism," because we want to understand what the value systems are, and it is fascinating how diverse um, um, people are. Um, about in this service, I just saw some of the data. 30% of the people who were asked is not a representative group at the moment. Yeah, it's but just people picked. Yeah, but 30% of the people believe that plants have consciousness. If you think this, yeah, they are concerned of this study group that we could harm and make plants suffer in what we do to plants. Yeah, and if you start from there, it is very difficult to run experiments on brain organoids which would not fall in a category where people would say, perhaps not. Yeah.
Yeah.
Um,
Okay. Well, that's very exciting. It's going to be interesting to see how this develops over the coming years. I'll put in the discussion section, I'll put links to some of your papers. Um,
yeah, best you link to our website,
yeah.
organoidintelligence.org,
Ah,
because
okay.
we have resources here, and we are continuously updating. There's a newsletter to subscribe to because we want to really form a community. And we already have initiated quite a bit. The White House made this one of the bold biotechnology goals for the US, and the National Science Foundation has an open call at the moment for Begin OI. It's called, so it's about engineering organs intelligence systems, and so it's a lot of.
Okay, great. Very exciting, Thomas. Thanks for taking the time.
It was a pleasure. Thanks for having me.
Okay, goodbye.